Okay, this we're picking up with sex-linked, still a monohybrid cross. Okay, we're just paying attention to whether it's a male or a female who's giving you that gene. Because in males, you only need one copy of the infected gene. This is going to be hemophilia, this is going to be color blindness, this is going to be male pattern baldness. Primary are the examples that we'll use for that. Okay, just a review. Okay, pedigrees. Let's talk about pedigrees. We don't breed humans. No, we do not. What we do is we go backwards and we try and figure out if the trait is dominant, recessive, if grandma was heterozygous, right? So we use males or squares, females or circles. Colored in are infected individuals. Okay, so if we look at alkaptonuria, these are people who actually pee black. It's a rare genetic disorder where the body can't break down tyrosine. So if you look at the back of like soda and candy and different things, it'll say safe for alkaptonuria or safe for AKU. And that just means if you have alkaptonuria, you can drink it or eat it because it doesn't have tyrosine in it. Okay? But it leads to a buildup of HGA and you pee black. Okay, so let's look at a pedigree for alkaptonuria. So in this case, dad is affected, mom is not. Okay, they have a son who is affected, marries a woman who is a carrier. Okay, how did we figure that out? How did we figure out that this person was a carrier? How did we figure out that this person was a carrier? Because they have a child that has it. Okay, we figured out that this person was a carrier because they have a child that has it. Okay, we also figured out that it's recessive because not everyone has it, right? These people don't have it, so it's got to be a recessive disorder. So that's one of the things we do with pedigrees is we go backwards. Okay, let's look at this person. How do we know if they are heterozygous or homozygous? We don't know, right? We know that someone gave them their big A, but we don't know if they got a little a from someone else unless they have children. Okay? So we'll do some pedigrees in class, and it's just kind of like a puzzle. It's just working backwards, right? Okay, dihybrid crosses. Let's get into it. We are not going to do it like this. It is too hard to keep track of. It is terrible. No, just no. Remember when I said every event is independent, okay? So it doesn't matter how many boys you have. If you have 5 boys, your chance of having a girl the next time is still 50-50, okay? These are separate events. So here we have for example, here's the parent of one, a tall green parent. See, big T times big G, little g. So this is a tall green parent of some kind with a also tall green, right? So we want to know what their kids are going to look like. Okay? Each time is separate. So we can do the big T, little t, and we say, okay, 50% of the kids will be TT, big T, big T. 50% of the kids will be big T, little t. Let's do that. Okay, let's just write it out. 50, 1/2 big T, big T, 1/2 big T, little t. Now, of the big T's, right? Let's just look at this guy right here, okay? Of the big T's, half of them will be big G, half of them will be big G, little g. Like this. So we have 1/4 of a chance of all homozygous dominant big T, big G. 1/4 big T, big G, little g. Okay, and you can keep going. So pause it and keep going, right? This is going to be 1/4, but you just have to follow this big T, little big G. Okay, that's what this one's going to be. What's this going to be? 1/4, because 1/2 times 1/2 is 1/4. Big T, big G, little g. So 1 to 1 to 1 to 1. Okay? We're going to do these in class. You're going to do it a bunch of times. Do not panic. Let's do it again. Let's use big A, little a, AA. Let's use B's. Okay, if you do a Punnett square, you know that this is going to be 50-50, right? Let's go back up here, right? Instead of T's, we've got A's. It's going to be 50-50. Okay, so let's look at the 50-50. Did I make you a? Okay, so 50, 1/2 will be big A, little a. 1/2 will be little a, little a. Do this Punnett square first. Okay, so we already know that. Now, of these guys, do another Punnett square here. 1/2 will be little b, right? Just look at this Punnett square. Don't worry about that anymore. We already did that one. Make a Punnett square here. 1/2 will be little b, 1/2 will be big B, little b. Now, you just multiply them, right? 1/4, 1/4, 1/4, and follow along big A, little b, right? And I may ask for phenotype, so you may have to describe to me this, whether it's a certain flower or a certain thing, but it's not that hard. Okay, let's do another one. Yellow, smooth phenotype here, okay? Of the smooth, we have big S. So we did this cross, and we found 3/4 are going to be smooth. 1/4 is going to be wrinkled, okay? When you're looking at something like this, I just put S and a mark because it doesn't matter. I don't care. I didn't ask you in this case for genotype. I just want to know how many are going to be smooth. So when you do an S with a line, that means it's big S, or big S, little s. It doesn't matter, right? 3/4 of them are going to be smooth. That's all we care about. Here, 1/4 of them is going to be wrinkled. That's all we care about. Okay, now, same thing for yellow and green. 3/4 of them are going to be yellow. 1/4 of them are going to be green. You add this together. 3 times 3, 9. 4 times 4, 16. Okay? Yes? So we have 9 that are smooth and yellow, 3 that are smooth and green. So the ratio here is 9 to 3 to 3 to 1. We got it? Okay, let's do another one. Now, all of a sudden, I'm going to ask you for genotype. This is harder. Now you got to do 3 of these because all of a sudden we care about this one. But we're just going to do the same thing, right? Okay, let's do our R crosses first. Let's look at our R's. Big R, little r, big R, little r. We did a Punnett square. Okay, so 1/4 will be big R, put it here. 1/2 will be big R, little r, put it here. 1/4 will be little r, put it here. Okay, now let's do the next Punnett square right here. 1/4, 1/2, 1/4. Okay, now here you go. Now we have these. Okay, it's just harder to keep track of. So you can do the same thing here, right? We already know. It's just now you have to keep track of that heterozygous. It's just a little bit uglier, a little bit more to keep track of. Okay? Yes, it's going to be fine. Here is yet another one, right, with the R's. Same scenario. This is looking at the ratios. We're just fixing this 1/8 and making it all 16. So we can do 1 to 2 to 1 to 2 to 4 to 2, right, if we want to do a ratio there. That's all that extra math is. It's just putting everything, it's just fractions, right? You're just putting everything with a common denominator of 16. Okay, a few more. We're almost there. Okay, sometimes there's multiple alleles for a single trait. Blood type is one of those, right? You can be O, you can be AB, you can be A. Another one of those is multiple alleles would be rabbits, okay? So brown fur is dominant to chinchilla fur, which is dominant to Himalayan fur, which is dominant to albino fur, okay? So this is how it goes in like order of dominance, okay? So of course we're going to breed some of these guys, okay? Here's what they look like. Aren't they cute? Look at the little flappy bunnies. Okay, so let's say we want an agouti which is, wait, which one is an agouti with an albino? Okay, so I think that's chinchilla in this case. Okay, they're just calling it something else. So we have big C and then a CH for this one. A C and a CH. And albino is little c, little c. You just have to read the problem and set it up correctly and then go back and see what the color is. It's not harder. It's a single monohybrid cross. It just, you have more than two choices. You just have to figure out which one of these is dominant. This is the same thing we're going to do with blood type. I'm giving you one. Here's another one with chinchilla versus Himalayan. Why is it doing, I don't think I want to do that. Okay, yeah, I guess I do. Okay, Punnett square, looking at a chinchilla by a Himalayan. So that's a chinchilla and a Himalayan. These two got together and we want to know what kind of babies they're going to have. Pause it, think about it, do it. And, you know, we can see that there's a chance. There's really no chance of having a white, right? Because that's not going to work. You really, the only way to get a white is to breed a white or to breed two Himalayans. But we're breeding a Himalayan and a chinchilla. So our choices are obviously to get a chinchilla, okay, or to get a Himalayan. See what the ratios are for that. Just write it out. There are also lethal alleles. So in cats, the heterozygous for Manx are normal house cats. This heterozygous are Manx. And if you have both of these lethal alleles, the homozygous dies. So if you cross two of these Manx, right, your offspring is 2 to 1. What? Right, because these guys died. It creates a weird ratio because one of them just doesn't survive. So you never get an offspring like that. That's just, these are like things to note. I will tell you if there's a lethal allele. We are not doing trihybrid crosses yet. I would not give you one on a test. We will probably do one in class. Crossover, we are going to talk about this later, but what we're doing now is assuming that there's no crossover at the moment. The process of crossover does occur, but we're assuming that that is not happening. Okay, also epistasis. So there are some times when you have to have two genes to get a color. Okay, can you think about that? Like you have to have multiple genes, right? If you think about eye color, hair color, it's not just one gene, it's multiple genes. So when you do that, we will do it, we will still not do it like this. I'm just showing you like this so you can see what the ratios are. It would be a dihybrid cross, but you're including epistasis. So this ending section, right, when we do a dihybrid cross, will just look a little different because you have epistasis. Okay? Don't worry about it. I will give you one in class. Okay, and that is all of chapter 12.